These are the untreated control, data from the untreated control. The blue dots are the optical stimulation, and you see a slight shift to the right in this. And the yellow dots are doing the same thing with electrical stimulation. This is the paradigm that Tessa Gordon has used for many years now. And you can see that whether we optically activate them or electrically activate them, we get an enhancement of the axon regeneration. Median lengths are much longer. I want to point out that they're hikers compared to exercise. Exercise makes much longer axons. But also, these guys were exercised 10 times, and the rest of these were only stimulated for 1 hour, one time at the time she did the surgery. So we think that the conclusion here is a single treatment with the optical stimulation. It does facilitate axon regeneration, and this is consistent with our hypothesis that that it's the activity of the neurons.
Can you stimulate after the injury?
You could.
Yes. So the optical stimulation, if it's after the injury, can that enhance?
We have not done that one, Gary, but I have done with electrical stimulation. It doesn't matter whether you stimulate before or after. For the electrical stimulation, you get the same effect. Now,
but the therapeutic implications are very good if you can intervene after the injury.
Yes, yeah, absolutely right. And that's one of the experiments Laura is going to do in the future. Okay. This summer we had a wonderful young guy in the lab named Sean Doherty. He's the one in the middle here. And he and Laura did these experiments together. It's the same paradigm. You did LED stimulation, but in this case we cut the entire sciatic nerve and repaired it. And then as a different outcome measure, we stimulated either electrically or optically above the cut 2 weeks later and measured EMG activity evoked in the tibialis anterior muscle. And so we just measured the amplitude of the compound muscle action potential 2 weeks after in this case it was 3 weeks and 6 weeks after. And this takes advantage of the fact that only 40% of the motor neurons, at least by our estimation, express channel rhodopsin. And therefore, what we would expect is what we saw here in intact animals, that if you activate the muscle electrically, you get a much larger response than if you activate the muscle optically. Because in this case, you're only activating 40% of the neurons, and presumably in this case, you're activating 100% of the motor neurons. If you cut the sciatic nerve and repair it, you wait 3 weeks, you get this really wimpy response here. And so we expected that with optical stimulation for an hour, we would only activate these motor neurons and only enhance the regeneration of those motor neurons. So that what we would expect, looking at evoked potentials later on, would be evoked potentials that were about the same size, whether we did them electrically, whether we tested electrically or optically. But if we did electrical stimulation and we activated all these motor neurons ahead of time, then we would expect this kind of a ratio in the optical versus electrical testing. And with one exception, that's what we found. That this is 3 weeks and 6 weeks afterwards. This is with optical treatment. They got an hour of optical stimulation, and you see that we activated a much smaller, we produced a much smaller potential, but whether we produced that optically or electrically, we got the same thing. When we electrically stimulated, we got 3 weeks, we got the expected result. We got something that looks an awful lot like this, suggesting that we had enhanced the regeneration of all, or a lot more of the regenerating axons, but we could activate more of them with electrical stimulation than we could with optical stimulation. And it all went to hell when we looked at 6 weeks, and we had some sort of reversal of the process. And if I really wish I could explain it to you exactly why we got this particular outcome, but I'm going to punt on that and say we just have to do more experiments. So these data are again consistent with our hypothesis that it's the activation of the neurons that's causing the enhancement of the regeneration. Okay, so we're almost everybody's going to be really sad if I let you go early. Okay, so the last part here is really the newest thing that we haven't really even done anything about, but I just couldn't resist putting in these pictures of Lenny Kravitz and Shakira. Because if we can activate, our hypothesis so far has been supported because we activated the neurons, but a crucial test of this hypothesis is we can silence the neurons while the exercise is going on. And our original intent was to use the halorhodopsins to do that, but I think the dreads are going to be a lot better. So what is a dread? It's not this. You probably all thought it was, but there was a subtlety in the spelling. Okay. And DREAD stands for Designer Receptors Exclusively Activated by Designer Drugs. And I couldn't resist putting more text on here because there are so many cool abbreviations in this field. Right? These used to be known as RASLs, receptors activated solely by synthetic ligands. These are basically G-protein coupled receptors that have been engineered to have no constitutive activity. That is, when you express them in mammalian cells, there's no endogenous ligand that's known to these receptors. But they will respond to a designer drug, a synthetic drug-like compound that, at least in theory, has no off-target action, meaning it doesn't activate endogenous receptors. And right at the moment, we're safe because the field is so new that nobody knows whether the clozapine N-oxide has any endogenous activity, but that's our story and we're sticking to it. And you can get these in both excitatory and inhibitory forms. Excitatory forms will excite the neurons, and inhibitory forms will silence the neurons. The clozapine N-oxide stimulated inhibitory dreads, called HM4DI, have been shown to activate G-protein inwardly rectifying K channels, GERKs, another great abbreviation, and induce neuronal silencing. The thing that is making these possible now that probably wasn't possible 6 months or a year ago is that these are readily available in different viral constructs from the University of North Carolina virus board. They're relatively cheap, and we'll see, right, whether it's easy for us to go through all the stupid administrative hurdles that we need to get them. But this is work that's going to be done by Kristen Sanders, who we're very excited just joined the lab. We will inject pre-dependent viruses. I'm showing it here in the sciatic nerve. We're not sure whether that's the actual route of administration, but we're going to try to use these viruses to infect neurons. They'll infect neurons in presumably via retrograde transport, and we can further refine the neuronal population that they infect by using the Cree models that I described before, where we can use chat Cree or Advillian Cree mice. We then can cut and repair the common fibular nerve the way that I've shown you over and over again today, and exercise them in the presence or absence of CNO. And this, if we silence them, and we have ways to go to prove that we've silenced them, then we can measure axon profile lengths, look at muscle reinnervation, and really we could even start looking at some of the behaviors, because we could, if we can silence afferent axons, for example, this would be a way that nobody's been able to do before to knock down specific populations of afferents. So that's what I have. We think it's all about the activity, and I'm happy to answer questions.
Very interesting. So in those experiments in which you do the optical stimulation, did the axons that have the cellular option were the only ones that were regenerating faster, or the ones that did not have the cellular option also regenerated?
We think based on we think based on the compound muscle action potential measurements that it was only the activated one. But clearly we have more experiments, meaning Laura has more experiments to do on that. We'd like to look more closely with retrograde labeling,
cells of motor neurons, both injured and uninjured, in order to calculate action.
Well, that's why we're hoping with the new mice, where we can look at chat-free or ad-villain-free, we can look at smaller populations. And if we activate them at different light intensities, we can even refine this even more, so that we could then, it's going to be a matter of how many ways do we look at it, do we see the same outcome every time? And so far, it looks like it's only the ones that are channel rhodopsin positive that are enhancing the growth. Yeah, Don.
The compound action potential, these are old studies reconstructed from peripheral nerve, are dominated by the larger fibers. For example, 70 or 80% of your amplitude can come from 10% of your fibers. And you demonstrated that the slow conducting axons were activated preferentially by optostatic stimulation. So could there be an interaction effect here where you're seeing primarily slow fibers and not the larger axons?
It could be. These
But
recordings
what you do with electrical, you see the larger ones.
Yeah, it could be. These are recordings that I showed you today were from tibialis anterior, which is like 90% type FF motor unit,
all right.
and not even really any slow. But that doesn't mean that there isn't a size gradation.
But just as an aside, the people that are looking at the prosthetic control, one of the problems has been how to stimulate muscle. And this idea of having a way of recruiting small axons prior to big ones, which is a very natural kind of activation by the nervous system, could be a tremendous breakthrough for these people. And we think about that contribution as well.
But we agree. We think it's really cool. Yeah, if I didn't portray that way, it's really cool. It is a bit of a breakthrough, yeah.
Have you thought about using the dreads rather than the channel rhodopsin for activation of those motor neurons? Because from a therapeutic perspective, it seems like it's
Yeah, I've thought about.
invasive.
Of course, I've thought about using the dreads because I like all those initials. And the problem with the dreads is it does take about a half hour to turn them on with the drug, but then the drug seems to last for like 8 hours, and so it would be really hard for us to compare the exercise for 8 minutes to something that lasted for 8 hours. It's perfect for inhibiting it, and in fact, an obvious control experiment would be to inhibit for 24/7, right? Just keep giving the drug overall. So I'm not sure I can, I'm open to the idea of using excitatory dreads, but it's a little scary.
Why do you think that androgens should modulate the effects of exercise on nerve regeneration? And as a related question, are there differences in human patients and gender differences in terms of the frequency of peripheral neuropathies that might correlate with the differences in androgen levels?
Well, I'll take the second one first. It's easy. I don't know. I don't know if there are or not, and frankly, we don't know whether these sex differences extend to humans or not. Dale Senglow, my collaborator, has a long history of working with pelvic floor motor neurons that are androgen sensitive. That's why we sought him out, because he kind of knows what he's doing and we don't. And he's convinced me that we should stop looking at serum testosterone and things like that, because this may all be a local effect. And why we think, I'm going to get to your first question. Why we think that the androgens and the exercise may be connected is that maybe the exercise, it has to be related to whether we think androgens are upstream from BDNF or downstream from BDNF. And we think they're upstream, and that we think it potentiates the effect. That one of the effects of the exercise is to make more androgens, either by synthesizing more testosterone or blocking aromatase. So one of the experiments that we clearly have to do is look at aromatase activity in exercised animals. When people have looked at muscle aromatase in female rats exposed to a paradigm similar to what we found, they found a tremendous drop in aromatase. So it would suggest, that's why Kevin proposed the experiment in the first place. So it's a long answer to, I don't know. Yes, Sean?
Comment first with regard to Randy's question and your answer. It seems to make sense to me that there'd be more local actions because when you exercise various muscles, it's those muscle mass that gets bigger. And so maybe there is some local mechanism of now, to the observation that after 6 weeks, after the optical activation experiment reverses, I think that'll parse out when you do your chat-free versus your adrenaline-free, because with optogenetics, if you're activating the smallest fibers first and you've got mixed sensory and motor, you're activating pain afferents at the same time, whereas with electrical stimulation at low threshold, you're not. So that's a factor that's different.
Yeah. I thought you really showed remarkable restraint. I thought you were going to talk about metabolism.
No,
No?
talked about that.
Which are, it's an in-joke. Sean thinks that we should consider, if we activate small diameter afferent neurons, that some of them may be what he calls metaboreceptive and therefore might sense that the animal is exercising. So it's actually a really good idea, but we haven't quite figured out how to study it yet.
Have you looked at how a history of exercise would change this rather than like, so exercising before the injury rather than after?
Yeah. One of the criticisms that has been, one of the concerns that's been raised over things like grant proposals is that you have to do this after the fact, otherwise it has no therapeutic potential. But there are 3 or 4 papers in the literature have done exactly what you're suggesting, Carl. They exercise the animals for a couple of weeks, and then they cut and repair nerves, and the axons grew better. And you could make the argument that, well, am I going to exercise on the thought that I might get a peripheral nerve injury? And some people might. Yeah, Sean.
With regards to keeping animals in cages versus the amount of activity they might normally do in the wild, is it possible that really when we look at our animals, and this is for all various kinds of studies, that they're sort of morbidly obese or inactive, and that when you do things like exercise or stimulation, you're actually more or less trying to direct them towards their normal level of activity? And how do people look at it?
I don't know. I thought the way you were setting the question up, you were going to ask me the question that I've been asked before and still don't have an answer for, which is, if we exercise them every day on a treadmill, are they more active in their cage afterwards? I mean, Bill is using some technology he's developing for you to try to develop an activity monitor cage so we can test that with, because I don't know. I can tell you that the BDNF knockout mice are really, the systemic BDNF knockout mice are really interesting because they get really heavy really fast. We've had 50-gram mice, and the usual mouse in our lab is about 25. And so you put them on a treadmill and they lose weight, but even though they don't have any BDNF. So yeah, you could be right. Other questions? Thanks. Great.